Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Joanna Smart from Business Review, and I will be your host. It is our pleasure to have Vironova with us today, who will be discussing automated purity and integrity check of gene therapy vectors. Today's guest speakers are Dr. Josefina Nilsson, Head of EM Services, and Martin Reiner, Strategic Development Manager. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. We'll allocate some time at the end of the session to address any questions or thoughts that you may have. If you click on the green resource list widget, you will find a PDF from our speakers to view or download. Please use the yellow help widget if you require any assistance, and you can move, resize and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Josefina.
Thank you very much, Joanna, for the introduction. So today, Martin and I will talk about automated purity and integrity analysis of gene therapy vectors. However, this approach can actually be used for any sample containing whole virus particles. But first, let me give you a short introduction to Vironova. Who are we? Vironova is a Swedish company located in Stockholm, and we are offering unique services for nanoparticle characterization and virus clearance testing, together with hardware and software for advanced electron microscopy analysis. To be able to do this, we have, or we need to have, I would say, experts in interdisciplinary fields, like electron microscopy, image analysis, programming, mathematics, and structural biology. I can actually go on and on. But by combining the expertise we have in-house, together with the in-house developed software, we are offering analysis of nanoparticle as a services, which is sort of the base. However, after discussion with our partners and clients, we have understood that they would benefit from being able to do some of this analysis in-house. We have therefore, together with the long instruments, developed a tabletop transmission electron microscope, which we call the MiniTEMT, which you can place in your ordinary lab settings. So, why use electron microscopy at all? I would say that is because particle characteristics like the size, shape, purity, actually the overall morphology of your product can often be directly correlated to efficacy and safety. So electron microscopy is a great technique to really get to know your product or to understand your design space. Like what happens if I change my formulation or purification process in this or that way. So for all of you that are working with viruses or proteins, you know that the process to get your products is quite complex and that there are a lot of parameters and steps to keep track of. Like aggregation due to unsuitable up or downstream conditions, the trade-off during purification, you want to get out as much pure product as possible, meanwhile not losing too much of your material. And of course, to make sure that your particles have the right morphology and are intact also over time. So let me give you a few examples of what I mean. Here we have 3 different AAV samples, the adeno-associated virus. To the left, you can see an image of a sample with a really nice looking AAV particle. The background is quite smooth, so there is not that much debris. But we can, however, see one unwanted structure, which is the proteasomes. In the middle, you have another AAV sample with much higher background, so you have a lot of small background debris. And you can still find the proteasomes also in this sample, as well as large aggregates, this one around 500 nanometer. Then we come to the right-hand side, where you can see a really nice looking AAV sample. Not much background debris, no proteasomes, but you can see confirmations of what we call, I would say, duplets and triplets. You can, of course, argue that this might be due to the stain that we have added when performing negatively stained samples. But if you also see them in cryo-TEM, cryo-electron microscopy, one can expect that these structures are also present in the solution. During formulation development, if not before, you want to find out your design space. What happens to my particles if I change the formulation in this or that way? Here we have one adenovirus sample in two different buffers. The one to the left-hand side seems to preserve the structure very well. You can clearly see the icosahedral symmetry of the particles, as well as the penton fibers in the fivefold axis. And you can also see the particles of single entities. So it looks really, really good, actually. However, on the right-hand side, you see the same sample in another buffer, which seems to not be optimal. You can see that the particle starts to break down and also start to aggregate. Also this one around 500 nanometer. So the one to the right is probably a buffer that you would not like to continue with or that needs further tweaking. You can, of course, also analyze your purification process. How does my product look before and after certain steps? Or compare different purification methods. Here we have an example of a picorna virus-like particles purified with two different methods. On the left-hand side, you can see that they are not being able to get rid of the baculovirus, which they used as an expression system. There is still quite high background of small, what seems to be protein-based debris, and the particle integrity also seems to have been negatively impacted. While using the sucrose gradient shown to the right, they got rid of the baculovirus, but they still have some proteasomes in the samples. So, and also the integrity of the particles looks much better here. So pros and cons that needs to be further analyzed. So there is a lot of parameters that can influence the quality of your product, and which can be monitored using electron microscopy. So after the poll question session that will start right now, Martin will let us know more about the MiniTEMT system and how it can be helpful with all the automation that has been built into the system. So I give the word to you, Joanna.
Thank you very much, Josephina. Okay, so it's time for a couple of polls. Audience members, if you could please select the answer that's relevant to you and click submit. The question is, where do you think innovation is needed in the biomanufacturing technology sector? In analytical methods able to link product understanding and process control, analytical methods for submicron particle characterization, disposable downstream and upstream technologies, continuous technologies, or other? So where do you think innovation is needed in the biomanufacturing technology sector? Analytical methods able to link product understanding and process control, analytical methods for submicron particle characterization, disposable downstream and upstream technologies, continuous technologies, or other. If you could choose the answer that's relevant to you and click submit. So the results are, 30% say analytical methods able to link product understanding and process control, 40% for analytical methods for submicron particle characterization, and then 10% each for the last three answers there. Now it's time for the second poll. So the question here is, based on your perspectives on state-of-the-art practices for characterization of biotherapeutics, please tick one or more of the following items as to their need for development of emerging technologies. So aggregation, process impurities, protein particulates over 1 micron, protein particulates submicron, identification of sequence variants, glycoanalysis, morphology, and particle integrity. If you could choose the answers that are relevant to you and click submit. The question again for you is, based on your perspectives on state-of-the-art practices for characterization of biotherapeutics, please tick one or more of the following items as to their need for development of emerging technologies. So that's between aggregation, process impurities, protein particulates over 1 micron, protein particulates submicron, identification of sequence variants, glycoanalysis, or morphology and particle integrity. So I will now go to the results. So here we can see a split between aggregation and process impurities at 68.8%, and then quite a varied response there from the rest of the answers. So thank you very much, everybody, for your input in the polling questions. I will now hand over to Martin to continue the presentation.
Thank you, Joanna, and thank you, Josefina, for the colorful introduction and for explaining the setting in which electron microscopy can provide insights in biopharmaceutical development and production. Now we're going to talk about the MiniTEM and the use of this system to answer some of the questions we have discussed previously. But first, some basics. MiniTEM is a highly automated transmission electron microscope that provides a spatial resolution of about 1 nanometer. And this is sufficient for most applications that we've talked about so far. Sample preparation follows established standards using, for example, unstained preparation, stained preparation, or thin sectioning. It has all the tools available for easy manual exploration of samples, as well as unsupervised screening and sample analysis. Because of the automation, the instrument can be operated on a generalist level, which makes it accessible for more personnel. The instrument can be placed in a standard laboratory setting with no extra requirements of mechanical or electromagnetical interferences, which is usually the case when acquiring a full-sized electron microscope. The installation and commissioning takes a few days. So I'm now going to guide you through the automated procedures in which the system works in order to collect and provide meaningful and accurate data of a sample. And we're going to discuss how this data and output can be representative for the sample and ultimately the product being analyzed. On the slide here, you see a representation of a standard 3.1 millimeter grid, which is used to place the sample upon and subsequently mounted into the instrument. To investigate sampling variability and preparation variability, it's a good idea to investigate different areas on the prepared sample grid. 5 or 9 areas are usually enough to investigate variability or trends throughout the grid. You can tell the software to visit pre-configured waypoints with a certain pattern, or you can manually, based on a low magnification assessment, tell the software which waypoints to investigate. The system then screens these areas automatically using the inbuilt operational automations. Each waypoint will be visited and each area will be imaged in a structured way. This would take a few hours if this is done manually, which for an experienced expert would not be an attractive task. So the system moves from area to area and collects images at each area based on user configuration. Each image is processed and particulate matter such as the primary analyte, but also undefined structures of interest are registered and accounted for. This is done for hundreds of images with 10 of thousands of particles available for statistical analysis. For biological samples, with its diversity and complexity, the automated operation takes about an hour. Easier samples takes, of course, shorter time to process until convergence of statistical parameter estimations. For instance, using this procedure, it's possible to reveal hidden contaminants that might not be handled in purification steps. Here we have an example of an adeno-associated viral particle production, which also contains what can be interpreted as proteasomes. The structures are presented very well in the image, giving a low rate of uncertainty, and the proteasome expressions can be separated from the primary analyte, giving both a representation of existence, relative concentration, and size, giving a hint how to optimize the purification steps. As a comparison, the two conformations cannot be distinguishable using dynamic light scattering, as seen in the lower right size histogram. We also tested using nanoparticle tracking analysis as a method, but it did not provide any data at all in this case. The condensed statistical output can be used to quickly assess the sample composition. Deviating data can quickly be investigated using simple point and click on graphs, which will guide you through the right image for further evaluation. In this slide, we see, for instance, a deviating large registered object. Maybe, Josefina, you can elaborate a little bit about the possible causes and how this information can be interpreted.
Yeah, Martin, as you said, when working with viruses, you normally know the size of the virus that you're working with. So the peak, the virus peak, is in the histogram is not that interesting, actually. It just confirms that you have your product. It is actually, the other information that do not belong to your virus peak that could be of interest. And there is, of course, many different reasons for why the material would form aggregations or aggregates that we went through before. It could be due to the formulation itself, a lot of background debris that we seem to have here, that the particles start to break down, or that you had a step in your purification that was too harsh and stressed the sample too much. And of course, it could also be due to the sample preparation. So I would, in this case, use an orthogonal method like the dynamic light scattering or similar to make sure that these aggregates that we see also are present in solution. And if so, then I would go back and look at the images to see, what is the original, what is found within these aggregates. That might give me a clue.
Thank you, Josefina. Also then, when having registered the primary analyte and other non-wanted structural appearances, we can aggregate this information even more to provide a metric of purity. On this slide, we see on the left side the area ratio between debris and the primary particle, in this case adenoviruses, for two different samples. The box plot shows the necessity to evaluate the grid at more position than one because of the possible variation throughout the prepared sample. This metric can in turn be cross-correlated to the product efficacy or ultimately unwanted side effects. As a comparison in time consumption of performing this analysis, we have a sample preparation that takes about 15 minutes. The microscope usage takes in the MiniTEMT about an hour, which is automated, leaving the analyst to perform other tasks compared with a manual operation of a few hours to collect enough images. The online image analysis takes about an hour per sample in the MiniTEMT, providing about 60,000 registered particles. This is not at all a task that is feasible to perform manually, but extrapolated data shows that this would take or mean about 2 weeks of manual labor. So another application of usage that Josefina presented previously is the possibility to assess particle integrity throughout the production process. Biologic materials are delicate and all process steps involve shear stresses to the products. I'm going to go through two situations, AAV particle integrity and adenovirus particle integrity. Now, in this slide, the visual representation of different conformations of the particles are exemplified. For the AAV particle on the top, we see from left to right, first what is believed to be an intact particle. The next show heavily internal staining, possibly caused by a compromised structure. Also, special observations of doublet or triplet formations can sometimes be observed. The reason of this is not confirmed, but may be due to unoptimal buffer or temporal change of conditions causing a disassembly and consequent unoptimal reassembly. The adenoviruses show a gradual breakdown. The cause of this will be discussed a little bit later. So the procedure is the same as for the debris ratio estimation, with multiple sampling points over the sample grid, multiple images, and particle detection and classification, giving a condensed metric of the ratio of intact particles compared to all particles detected. Since preparation procedures of samples can affect the particle integrity, both stain selection and stain quality of grid areas are critical factors for accurate viral particle integrity measurements in transmission electron microscopy. 3 images acquired with MiniTEM are shown on this slide that shows representative grid areas with different stain quality of AAV samples. On the left, marked with a B, we see a grid area with poor stain quality where the stain has been dried out, showing a whiter background compared to the other areas, and particles lack the typical stain halo that is observable during optimal staining conditions. When we scan for viral particle integrity status in these areas, we see a clear correlation between stain quality and particle integrity, which is an unwanted dependency. However, over a certain threshold of stain quality, the results stabilize and a comparable measurement between samples is obtainable. Adenoviral particles are more sensitive to the effects of staining agents compared to AAVs. Here we have 6 different samples showing the same degradation of particle integrity when the staining embedment surrounding the particle is poor. The top left image shows a completely dried area, which is also represented in the yellow dots B in the graph. The correlation between the sample preparation and the integrity measurement is high, with a high value of interference. Also the case with the next image, C and possibly D. Over the stain quality measurement of 1, the integrity measurement shows a low correlation to the preparation procedure, and the interference is also low, which makes it a good observable. And as in the case of AAV, the measurement is comparable between samples. Now, for the AAV samples, we can distinguish between the intact form and the internally stained non-intact form, giving this a separate measurement, and also the ratio of doublet and triplet particles in the population. So we have here 2 samples with differentiable level of both intact particles, but also with a differentiable level of doublets and triplet formations. If one would keep track of these parameters, the design space could be further detailed and the effect, if any, can be monitored. For the adenoviral samples, the preparations of sample B, C, and D are disqualified automatically, but the samples E, F, and G show a comparably different integrity as well as internally low variability. The reason for the adenoviral integrity variation between samples can, for example, be linked to stresses on the surface proteins, such as the pentones, caused by filtration or buffer composition.
So to summarize, I hope that we've been able to show you that we are trying to automate this system for also people that are not experienced microscopists to use. And that we really think that by having a MiniTEM at your site, you would be able to do detailed characterization in your early development, which can then support both the formulation and production. And by identifying critical and measurable parameters for your products in early development and by using them throughout the whole process, you will be able to develop robust processes as well as high quality products. And don't forget that we will be there helping you all the way. Depending on your needs, you can use our services where we take care of the whole process. Would it be that you already have an electron microscope at your site? You can use our image analysis software, VOS. And would it be that you would like to do all this in-house in your own lab settings? MiniTEM is the way to go, although you might not be an experienced microscopist. Thank you very much for listening to this webinar. And you can, of course, also contact us directly if you would like to by phone or email.